Today oral presentation title is like this, about food and biopharmaceutical science. My title is Discovery of Novel Bioactivities Phytochemicals from Neighboring Plants. I plants especially maybe is Vitex is rotundifolia and almost sample is Vista species. I present is like this is as follows this research area. My name, our is lab is pharmacognose lab. We research about the natural product phytochemicals. So we targeted research on the functionally is a natural product to discover active phytochemical derived from as plants, especially in Jeonnam regions is plants. So also is establishing various bioeffects evaluation system and using the replication technique by using high SMS molecular networking system. So first one is phytochemicals. After collecting natural product, we conducted LC-D8 mass analysis in the isolation, and also is purification of active candidate phytochemicals as compound. And part two is analysis. We established the SMS-based replication and the active candidate and the chemical profiling, the simultaneous analysis for standardization. And also as we try, is evaluating system is for bioactivity. So we target is whitening is anti-inflammation is antioxidant is but target in IBD is just we try is in vitro system. Natural product, is many is about the natural product. This is a bio industry. Industry is utility is a product derived from the animal and the plants based on the efficacy and the functionality. So, and the natural product is industry include natural medicine. We call it as a new botanical drug, 천연무신약, and the functional food and the functional cosmetic. So recently we researched about the functional cosmetic. We call it normal cosmetic, and also now we research is pet food. Yeah. So, and as you know, you can see this bio industry is grow domestically and globally. So the standardization, the concept is standardization has become very important, in natural product bio industry. So it's for R&D and is more natural medicine development, is functional food. So the standardization of technology information used throughout the manufacturing process, from raw material to product, for maintenance of consistent quality control, regardless of many various produced batches. So as a natural product material resource, we many conducted focusing on the plant source that growly in Korea naturally. So I selected it, especially Jeonnam region is a plant. So like this is a research, and I researched the samples. So I conducted southern forest, academic forest of Seoul National University, located on the Baegun Mountain in Jeonnam, Gwangyang. So, and also is now recently I searched my sample is near Suncheon, my home, because my home is you guys know about the Suncheon Bay International Garden Expo. Suncheon have many plant sources. So I recently is collected because my home is near the garden expo. So and also I like is collected in Gure. Gure have as a Jiri Mountain is very famous in Korean. So and also this is my is material my source. This is a list of is anti-cancer but target is target. So applicants was selected by searching based on pre-reported paper. Traditional report is okay. The target activity in so for example, I target eye cancer and anti-cancer list of is many various many target and also the list of yes, and the specific biomarker is all yes is reported not reported and we search it many is using many platform and the specific biomarker narrowing the scope the way high potential first one we target as a research reported or non-reported and patent because patent is very important in for development. So the phase on of our series for discovery for active phytochemicals is we based on the traditional activity guided isolation. Is many about this is a skill. So and recently we use molecular networking, the replication using LC, mass and recent metabolomics. So and we targeted the discovery of active marker and because marker compound for is important to the standardization. So and we also perform the chemical profiling. Simultaneous analysis is a method validation for standardization. It's like this is a figure. Phase 2 and 3 is we conducted target isolation. So this method, after correcting, we got the large scale curve extract and then we isolation, fractionation, separation by using automatically the various LSMS system. And then we elucidate is what is this structure? Is the elucidated structure, chemical structure, and by 1D and 2D animal. And the compound finally is by mass. And the end we just try a video is a bioassay. Our study is focusing the many focus on evaluating the activity biomarkers related to the inflammation and oxidative damage in target IBD. So and also is I should research the functional cosmetic because there is many requesting from many companies. So I also is a target functional cosmetics. I try the research is whitening is more antioxidant and development of functional cosmetics. So and also the global natural product social this is a web platform have many mass data. So we study is GMPS based molecular networking is using high resolution orbital mass. This is a state of in my lab. So and also ultimately aiming is at GMPS molecular networking is guided target isolation. Our is ultimately is our goal. So this figure like this. And also additionally we attempted investigate the activity by establishing LC based antioxidant evaluation system using DPPH-LC analysis. And also is pampa BBB method. Since last year, we try is molecular docking in silico system by measuring binding affinity. Just with now, we started and the binding affinity calculator was operated as Autodoc, and the visualization is expressed the PyMo and the Discovery Studio. So. I presented the natural product is contents that published in paper is during 1 year, recently 1 year by our lab. So I will present is first is BTEX rotundifolia and Bicep species, and one more step. Briefly. So the first sample is Vitex rotundifolia. This is Korean name is sunbigi. Sunbigi tree fruit. Its name is in Korean is manhyongja. Is used in the Korean medicine as a treatment for various diseases such as cold, headache, and chronic otitis. It's many use. The leaves and twigs of sunbigi tree have a unique scent, and they are used of varying material or to remove moisture. I collected. I first collected is gohung. Gohung is near Suncheon and have coast. It's a very clear and beautiful spot. As I recommend is going visiting. So during visiting, my child, my son, and I searched this is sample. Oh, what is this? This sample is very seen is fragrance is very good. So what is this? And I searched, I reported, studied, is reported paper. Is I think is a very good sample, so I select. So my title is including neighboring plants. Is I make it this is a name because during going for walk, visiting tour, I search the samples. So this is the sample is in our lab is extract and isolate is analysis and try bioactivity assay. It's good. I mean, I go on. So I selected like this, and sunbig has many chemical structure. Like you know, is chlorogenic acid is phenylpropylenoid type, and also flavonoid and iridoid type is many various is type is a structure. So first, our experiment is first we select the extraction condition. So in order to determine the extraction solvent, we took condition solvent was set is 80% ethanol is methanol. So yield and bioactivity is evaluated. Based on the yield evaluation and activity, we selected 80% ethanol is more good, more better. So this yield of leaf, twig, fruits evaluated at the highest. So it's got the large scale. The collect sample is extraction. The excluded fruit just we selected is a leaf and twig. It's performed to isolation. The fraction on performed by polarity. We got the many fractions, extraction and fractions, and try other further study. So 80% ethanol extract of 5 organs. So we first selected a sample and then we all part of a plant we collect and we try all organs, all parts, and among them we select. 80% extract of 5 organs is exhibit the stronger radical scavenging capacity. Yeah. The leaf, twig, flower extract is of sunbigi is significantly reducing capacity, is antioxidant activity. So in ABTS experiment used and some fraction have we confirmed we checked some fraction is good. And the 7 marker compound we target isolation is 7 marker compound for this is a project, this is a paper is target is analysis standardization. So we 7 marker compound isolated is obtained using multichromatic graphing method, including just 그냥 simple open column is a 저기 biomass is a lot of. So MPLC and the self-rep recycling analytical LC. This is the figure is just one page, but this experiment need labor and time. So the figure is very simple, but this is labor and time is a lot of. So anyway, is isolated is marker compound target isolation 7 is rechecking the mass and mass. Their structure is established by analysis of their spectroscopy data is based on the mass comparison in the previous study. Also, the structure was verified by observation, is calculated the data and the original experiment data is molecular weight is and the fragment is compared. We elucidated what is chemical structure. Yeah, like DS is a 7 markers is compound. This is just a simple is phenol compound and pennypropanoid and iridoid type is flavonoid. This is a 7 marker. So finally, the HPLC analytical method was successfully is established. And we also we got the main result of like this regression equation, correlation coefficients is a value. This is a 39 is good data as a purity and also LOD and LOQ. And also the established method was used to analyze the chromatography is a fingerprint. We call it as fingerprint of sample is Sunbig sample. Signal clearly expanded the chromatography profile of 5 organs samples. We established the method validation. So it's 5 leaf, twig, flower is fruit, and also it's a root. This is a tree, so we have a root. So we additionally try is the DPPH HPLC system. DPPH LC system is resync is novel. The useful is for just for rapid isolation, rapid screening, the identification. Earlier I mentioned the isolation using chromatography is need more labor, more time, more fund. So we continuously develop the rapid isolation, rapid screening. And as a result, compound 1, 2, 4, 7 is a very high antioxidant effect is a DPPH LC system. This is all, they are all 70% more. Just among them is compounds 4 and 7 exhibit strong antioxidant capacity with a reduction of a peak area. Yes. So we conducted as quantification of marker compound contents in all organs. Sunbig collected with differences of location. We first started collected Goong. I like it just personally. I like it's Goong cost. So, and the first is very good is data. So we try at that year, maybe it's June, collected, collected is Goong, and at that the same year is I had to go conference in Busan. Yeah, so in Busan is collected, and also is maybe summer is I had to conference Jeju, so it's collected, and also the Sinan. So this means South Sea is East Sea and West Sea, and then this is a region in Jeju. So as a result, the result I published this is a paper. It's about the Sunbige first paper. But as mentioned earlier, the standardization has become very important is concept in natural development because there is many reasons, many problems. So phytoequivalence, quality control, and the elucidation is active compound, active phytochemicals. There's many law is a law is needed is elucidated is all constituents, all phytochemicals. So I focus on the research in the standardization. So we secured the research fund and operated as Biomedical Global Center, and research is standardization of company to conducted is standardization research to support food and biopharmaceuticals R&D of by requesting companies request like these contents. Yeah. By further study, Sunbigi, I isolated is 46 phytochemicals, including 7 new compounds. 12 iridoid glycoside is isolated 10 quinic acid, 4 phenolic glycoside, and 20 phenolic acid flavonoid triterpene. First is next I research chemical structure type is iridoid. I focus on iridoid. So the structural elucidation was performed based on the like this spectroscopy, the 1D and 2D NMR and or acid hydrolysis. This compound, iridoid glycoside, and it means iridoid have sugar. So we have to elucidate is what is sugar, what is stereochemistry of character of compound. So we use like this is method. Iridoid was evaluated for inflammatory effect through inhibition of animal production and like this compound, some compound have good anti-inflammatory activity and the iNOS COX-2 and also we target the use HT29 cell line. So we measure the IL-8 production is a reduction. This is like a compound is have a good show the good activity. So we publish the phytochemistry like this including this data. So and also this is many is flavonoid triterpenoid like this is isolation and is measured bioassay, and the structure was determined based on the NMR and the CD and the mass is MTPA like for elucidate spectroscopy is character. So they have showed the many effects. So we conducted in on silicon docking approach also revealed the strong binding affinity. This protein ligand complex is active compound against we target the protein IL-8 production. Yes. And also we experiment ILA assay and in vitro assay and in silico is continuous simultaneously. So like this, we published the paper, and currently the three papers for patent application on technology transfer. Three projects have been is conducted. Additional research is being conducted on macular degeneration. This is a hot trend is functional food. And anti-cancer using quinine acid phenolic compounds. Since last month is September, we start a new project for cosmetic development. The development is name is R&D Rediscovery Program by Kiat. Also is we are preparing a study paper about the quinine acid phenolic glycoside as eyes and cosmetic and cancer. Also is other sample is similar is currently this is the same is the plant name is Osmanthus fragrans varieties is Aurantiacus is the name is 금목소. You the current this is similar. That's one more sample. This is a BCR species. The parliamentary alcohol procedural molecular networking was performed. Untargeted is SMS analysis. Our total extract is different organ. The same species, same genus name is have many species. We selected many species and we many selected organs. Three organs. The replication of SMS spectral data was conducted using feature-based molecular networking. So the pie chart suggests the flavonoglycoside contain a relative abundance of this sample is a flower. So subsequently, the flowers of this sample is in Korean name is 등갈퀴나물 is used to optimize extraction process and purification. We isolated, we got the one new compound and 23 compound. The verifies anti-inflammation property is active constituent. We target the molecule docking is binding affinity. The target COX inodes IL. We try as a target as anti-inflammatory. So also sample is this is name is in Korean is 벼룩나물. This is this sample also is growing widely in Jeonnam region. We started on some company request is R&D to me, so we started the sample. So this sample, this is ultimately our goal is like this is a paper title. So we first present is a molecular networking guided isolation strategy of new compound is from is a natural product. So summary is our research is a summary. Goal is a pharmacology phytochemicals, active phytonoble compounds from domestic plants. I mainly is conducting collecting in the Jeonnam region because it is my near. Our home is near. So through bioactivity guided fractionation, isolation, and LCMS plus LCMS based de-replication, and plus GMPs based molecular networking guided isolation. Our goal is development rapid isolation. Our law, I think, or discovered as a useful functional material is a very important point. Functional and to development product against the inflammation and the oxidative damage. And now I expanded is a target active point. And also marker compound are validated and quantified for standardization. So this is anyway, our research goal is like this. So our lab is a researcher. Thank you for attention.